Hello, everyone, and thank you very much for being here today. Very excited to have you join us for this special event. My name is Andy DeSoto. I'm the Director of Government Relations at the Association for Psychological Science. If you aren't familiar with APS, we are an international organization of over 30,000 individuals dedicated to advancing psychology across disciplinary and geographic borders. I'm really pleased to announce today's webinar, which is titled Clinical Psychological Science Through the Lens of RDoC. New advances and future directions. Before we begin, just some quick logistical reminders and details. So today's event will be 90 minutes long, which is a slight change from the initially advertised time, 90 minutes. As participants, your attendees, your cameras and microphones will be off. If you'd like to ask questions of our panelists today, please use the question and answer feature, which is at the bottom of your Zoom window. Our time for questions and discussion will be at the end of today's session. Now let's get on to the topic. As part of the mission of APS's government research funding and policy initiatives, we are very interested in sharing the latest and greatest psychological science, as well as the government science agencies that support and conduct that work. Today's webinar is focused on innovative research in clinical psychological science, and we're really excited to have brought together scientists doing cutting-edge research in this discipline with representatives from the National Institute of Mental Health, which is the US's lead federal agency for research on mental disorders. Here's what you can expect today. First, you'll hear an introduction to NIMH's Research Domain Criteria, or RDoC, which is a research framework for new approaches to investigating mental disorders, from Sarah Morris, who is Associate Head of NIMH's RDoC unit and the Branch Chief in NIMH's Adult Psychopathology and Psychosocial Interventions Research Branch. Next, you'll hear 3 research talks from psychological scientists Vijay Mittal, Adam Kujawa, and Anne-Marie McNamara. Our discussant for today's program is Uma Vaidyanathan, who is Research and Innovation Manager in the NIMH's Director's Office. As we have a full program to enjoy in the next hour and a half, let me pass things right over to Sarah Morris, who will say a few words of introduction to RDoC. Thank you, Sarah.
Thank you, Andy. And thank you, thanks to APS for hosting this webinar. We're really happy to be here today. Okay, so I'm just going to give a very brief overview about RDoC today. Talk a little bit about the rationale for RDoC and an overview of the RDoC structure. And our principles related to RDoC research. And then I will hand it over to the main event, which is the research talks from 3 outstanding investigators. So the first phase of the rationale of RDoC is really dissatisfaction among patients and family members with existing diagnostic structures. Anybody who has used the DSM or the ICD knows that patients may receive multiple diagnoses, and that although the DSM has allowed a decent level of reliability, it's not as good as it could possibly be. Oftentimes providers can't agree, and patients end up going through a very frustrating and sometimes prolonged period of diagnosis, misdiagnosis, re-diagnosis. And those diagnoses provide only modest information about clinical prognosis. Treatment remains a trial and error endeavor, with many people needing multiple different treatment approaches before reaching an efficacious response. Distress is a major symptom and criteria for the DSM, but it varies from day to day and hour to hour. And then our current diagnostic frameworks don't give good information about when should treatment end. And that's a difficult decision for clinicians. And then from the scientific point of view, for researchers, our current systems remain based on clinical symptoms and signs, which are difficult to measure objectively and reliably. Although the systems are devised for reliability, they are arguably lacking in validity. Although the DSM presents diagnoses as if they are valid scientific entities, the data increasingly shows that perhaps they are lacking in validity as measured in different ways. DSM disorders are broad syndromes. They are heterogeneous. There are high levels of comorbidity and high reliance on not otherwise specified diagnoses. Disorders are, as we know, disorders occurring naturally are dimensional, that there's not a bright line between healthy and sick, as is suggested by DSM categories. And so scientifically, it's important to look at that spectrum of health to illness and not prematurely draw bright lines and boundaries between health and illness or between disorders. And all of these weaknesses in the current diagnostic approach have contributed to the failure to establish biomarkers, because it's very difficult to find a biomarker of an illness that is so heterogeneous and overlapping with other disorders. So we were faced with a problem 10 years ago. Before RDoC was launched, we were faced with a problem that these diagnostic categories, which have allowed the evolution of the science of psychiatry and are essential to clinical practice. However, they were driving the entire research and clinical systems. Grant reviews, journal publications, clinical trials, regulatory approvals are all based on DSM diagnosis. And so the NIMH wanted to take a step back and move away from this sort of unwritten rule that NIMH funded research should only be guided by what's in the DSM. So the maybe bumper sticker version of what is RDoC is that the National Institute of Mental Health is not the National Institute of the DSM. And researchers should be free to explore new ways of classifying disorders in the hopes of achieving more precision in diagnosis and treatment. And so at that time, again, we sort of took a step back. What do we know about mental disorders? We know more than ever about behavioral neuroscience. We know that psychopathology occurs along neurodevelopmental trajectories. We know that there are bidirectional interactions with environment in both risk and protective ways. We know that psychopathology is dimensional, and that we know that we're best off if we measure and relate many aspects together: the psychological, the biological, self-report, behavior, instead of focusing on any one of those measures as the gold standard or the foundational. And so with the long-term goal of moving away from one-size-fits-all diagnoses toward dimensional transdiagnostic mechanisms, we implemented these strategic principles, trying to invert the clinical paradigm. Instead of starting with a mental disorder as defined by criteria and trying to understand the mechanism of that disorder, starting with what we know about normal, healthy, adaptive mechanisms, and figure out where things go wrong to result in pathology. Again, focusing on that normal to abnormal dimension, studying transitions to psychopathology developmentally, so to inform early intervention, using data-driven constructs jointly defined by behavior and cognition as implemented by neural circuits, and more recently adopting computational neuroscience approaches to understand those relationships that are complex and not apparent to the naked eye, but can be derived using computational approaches. The RDoC framework itself is embedded in a lifespan developmental trajectory, acknowledging various impacts, the various environmental factors along the lifespan, and thinking in terms of dimensional constructs that can be measured using multiple units of analysis. Again, with the interest on capturing that whole span of the dimension of a construct, and then looking at the extremes and figuring out how the psychopathology arises. We've had this series of funding announcements related to RDoC starting in 2012. These have resulted in over 95 grants funded under these specific announcements, but then of course many other grants that are funded that are related to RDoC but are funded under other funding announcements that aren't specific to RDoC. So it's a little difficult to quantify the RDoC portfolio at this point because any given research project can implement certain principles of RDoC. So there, again, it's dimensional in and of itself. There's no bright line between what makes an RDoC project and what doesn't. But these 95 grants are good sort of gold standard exemplars of RDoC. Another example, we were really gratified to see this big project funded by NIMH called the Aurora Project, Advancing Understanding of Recovery Under Trauma, informed by the idea that the pathological response to trauma can take many shapes and forms, and that it's important, again, to understand that heterogeneity across different types of trauma and different profiles of outcomes. Another example, an international example, psychiatric ratings using intermediate stratified markers. This is an Innovative Medicines Initiative funded multi-site study of social withdrawal transdiagnostically across schizophrenia, Alzheimer's disease, and major depression. Again, to look for transdiagnostic mechanisms related in this example to social withdrawal. And finally, just to talk a little bit about clinical trials, of course, ultimately with RDoC, our goal is to improve diagnosis and classification and leading to better, more precise therapeutics and novel treatment targets. So we agree with this paper where they said we see little alternative to the pursuit of transdiagnostic functional domains as the core targeted features of neuropsychiatric disorders. So I will stop there and thank you all for attending, and I am happy to hand it over to Dr. Vijay Mittal.
Vijay, you might be muted just in case you're not aware.
Sorry for the delay there. I want to thank everyone for this opportunity. I'm very excited and honored to be part of this panel and really excited about where RDoC is going too. And before I begin today, I just want to thank my lab and my family and the funding agencies, particularly NIH. It's been incredibly helpful to have their support during all of this work. So before I start talking a little bit about a big aspect of my research program, I want to frame the clinical population in question. The work I do focuses on the psychosis prodrome, and this includes young adults and adolescents that are showing signs that they're at risk for developing a psychotic disorder like schizophrenia. The signs can vary, but they look a lot like delusions and hallucinations. Negative symptoms, things like affecting affect, cognitive decline, social withdrawal. We use clinical interviews, and we have some really strong criteria that the field has decided on, and that allows us to collaborate in a reliable and valid way. But when I'm giving talks, for people outside of those that use that interview, I like to use these Louis Wain cat paintings to kind of make the point. Louis Wain was a prolific commercial artist in England, and he developed schizophrenia. And he continued to draw. And you can see here cats that he drew during his course of illness. And the prodrome, beginning to look at people at risk, would really represent this first line of cats, where there's something off, something odd that's going on that you can't put your finger on. So the teenagers that come into our lab are starting to maybe think their parents called their name, but then no one was home, or they think they saw something out of the corner of their eye. And we are interested in following people that have these experiences recently start, or if they're more longstanding, have them get worse recently in the last couple of months. And as we follow them over time, the criteria along dimensions of distress, frequency, severity, insight, conviction, they all increase. So somebody might start to put together that they're thinking that someone is home and they sense a presence, and they might link that to another symptom, and it might make them miss school the next day, and it escalates up this ladder, just like Lewis Wayne escalated, and his perceptions of normality changed to the point where we would consider that psychotic. And so when we use this paradigm, which was really invented and refined in the early 2000s, we're able to follow teenagers and young adults. And in about a year, 15% of them will go on to develop a psychotic disorder like schizophrenia. And in about 2 years, roughly a third of them. Since this work came out, the numbers have declined slightly, and we believe that's in large part due to the efficacy of some of the early intervention efforts. The great hope of this work is that we'll be able to stop psychotic disorders from happening. And there's lots of promising treatment work here. There's lots of benefits in finding people before they develop psychosis. I've listed a few here, but there are many, many more. And in addition, having these longitudinal studies where we can prospectively follow people in a period before many of the third variable confounds associated with schizophrenia research aren't there, like years of antipsychotic use and substance abuse and things like that, allows us a clear mechanistic understanding. And so there's been some major breakthroughs in the field of psychosis because of this work. But there's still some questions and challenges. We are not great at finding people who meet criteria for this. We use a lot of resources for that. So for the general population, we need a lot more work on screening and screening out noise and other disorders. And in addition to that, once we find people, only a third of them at best will develop the disorder. And so we can't just use blanket treatments. These treatments are expensive and they have side effects and sometimes, and there's a lot of other reasons. So being able to find who might develop the disorder among phenotypically similar youth is also a major priority. And then the last major priority is understanding what is driving this illness. We still don't have a great handle on what causes schizophrenia and psychotic disorders more broadly. And until we do, we're really not going to be able to develop the kinds of treatments that these patients and individuals deserve. So my solution to addressing these questions and challenges has been to look at motor behavior. And that might sound like a fairly arbitrary or random decision, but I assure you it is not. It turns out that several of the circuits that govern motor behavior are also highly implicated in the pathogenesis of psychotic disorders. And in fact, when you follow motor development and look at abnormalities in people that develop psychotic disorders, it follows what would be predicted by a typical diathesis stress model. So we know on the far end that when people develop schizophrenia, independent of medication, they show a variety of abnormal movements. We're going to talk about several of them today. We also know that children, infants in fact, that one day will develop schizophrenia later on in adulthood, show motor delays as well as characteristic abnormalities. Some of the work from my doctoral mentor, Elaine Walker, has been a landmark in this area. And I'm going to show you some examples here of what I mean. So this is a little blurry, forgive me, but you can see here that there is a withered hand. I'm just going to pause it there. Oh, when I pause it, it goes away. All right, let me. So this is a type of dystonia, anathetosis, where there's a muscle spasm, where an agonist and antagonist muscle twist. And that's something we see a lot in adults. This child is using a highly asymmetrical crawling pattern. So there's issues with motor laterality here. This child only uses one hand to manipulate the object. There's poor fine motor skill development here. At a year, they're not able to manipulate the package. They're using a pincer movement. And here at 8 months, the child is not able to right themselves. And that speaks to a paucity of movement. So Elaine's research compared videos of people, childhood movies with schizophrenia. They had brought them in and compared them to community controls and found motor delays, delays in milestones, and these kinds of abnormalities. What was so interesting with this research was that by the time that these youth were later in childhood, right before adolescence, these kinds of signs were no longer visible. And that's not because they went away. It's because people eventually do meet milestones. And so that is no longer a useful index. So we know the vulnerability is there because it's there once people are psychotic and it's there from birth. But we can no longer see it very easily with the observational methods that we used in the Walker videos. And so my idea was that perhaps in these teens and young adults that are starting to have emerging psychosis, we might be able to see who is going to develop the disorder early by seeing if these signs kind of start to re-emerge or become more obvious, like we see them in psychotic disorders like schizophrenia. So I got very excited about doing this work and was all ready to launch my career in 2010 when I first became an assistant professor, and RDoC came out, and they didn't have a motor domain. And at the first pass, it also didn't look like they weren't taking development very seriously either. And so that was disappointing for me. I was very excited about RDoC. But I have to say, and I'm not just saying this because all of the RDoC people are here, that they're incredibly responsive. My team and several others, wrote a lot about these issues, and the RDoC team wrote back, and there were workshops and development, a lot of development. This whole RDoC thing is labor of love, and there are a lot of smart people nurturing it, and I'm really excited to say, they became a motor domain, and it's perfectly lovely. I'll show you lots of examples in a few minutes. And there was just recently an environmental workshop and a developmental workshop where we worked to build models that would better highlight development. So another really good thing about motor behavior particularly is that it's very circuit-centric. Understanding circuits in schizophrenia and motor in schizophrenia overlap quite well. So for example, basal ganglia circuit abnormalities will result in that athetosis I showed you, but also some slowing. So you see some rigidity and tremor here, some ballistic head, neck, trunk movement here. Cerebellar circuit abnormality, which is also highly implicated in psychosis, will result in clumsiness, as well as a variety of sensory motor integration abnormalities, deficits in motor learning, and some other things we're going to talk about in a few minutes. And then there are these corticocortical motor circuits that are involved in more complex higher order movements like gestures that are linked together with cognitive and language processes. And all of these are impacted in psychosis. And so it's exciting because we can look at specific motor behavior and get an idea of what's going on in the brain and understand potential pathophysiology and also look at clinical targets as well. So another part of RDoC that I mentioned that I'm excited about is how well it adapts to development. And I'll use more motor examples here. So I mentioned when I was showing you Elaine Walker's videos that when the children get a little older, it's difficult to use something like milestones as the big marker. And that's because everyone develops milestones eventually, or most people do. And so it's no longer an easy thing to detect. Here you can see, by age 4, even people that are quite delayed, eventually develop these things. So how do you, index that vulnerability later on? Well, you might consider a different unit of analysis. So, maybe the videotape um, observation method doesn't work anymore, or looking at hospital records or parent report of delayed milestones. So you might try a new unit of analysis, and we have been finding that that's really useful. So this example isn't exactly like an earth-shattering new unit of analysis. It's more of an experimental paradigm where children interact at a lunch table. We're still using motor coding here, but we're looking at fine motor movement this time. So this is from Jason Schiffman's work. He looked at the Sarnoff-Mednick Danish high-risk data and took the videos that were there of children eating lunch. These are 11 to 13-year-olds. Both of these children have a parent with schizophrenia. The child on the right, the young man, develops schizophrenia in adulthood, and the young woman on the left does not. And you can see here, when you look at fine motor movement, now there's an obvious deficit. And you wouldn't have been able to pick up on this in childhood because fine motor skills were still in ascendance. But here in early adolescence, late childhood, it's quite obvious. You can see he's using his whole torso and his upper body to use the fork and the knife and spoon. And so the idea here is that you can get at the same vulnerability with developmentally appropriate assessments or units of analysis. So for example, we see clumsiness in children that go on to develop schizophrenia. But eventually, adults are much less clumsy. And so how do you get at that vulnerability? Well, we use an instrumental assessment paradigm, which would fall under the paradigm unit of analysis. And specifically, it's a highly sensitive scale that allows us to detect extremely minute sway. So the participant is required to just stand there in this particular condition, and you can see her you can't see with your eyes that she's moving, but when you're balancing, you're actually doing lots of tiny little corrections based on sensory input, and it's challenging your cerebellum. It's actually kind of a miracle being able to stand. And in schizophrenia, we see wide sway areas here. And so we're able to parameterize that. And again, it wouldn't be possible to get a young child to stand on the scale, but similarly, flipping it, you could get an adult on it and you wouldn't be able to see clumsiness. So when we use paradigms like this, we're able to see high-risk individuals have a much wider sway area. And some of the follow-up work in my lab has linked this to cerebellar circuit abnormality, structural and functional. For the purposes of these slides, UHR and CHR are acronym that mean clinical high risk or ultra high risk. So that's the prodrome. So the first circuit that I want to briefly talk about, I'm going to give you an example or two of each circuit, is the basal ganglia circuit. And this is central to the dopamine hypothesis of schizophrenia. It's believed to be highly implicated in positive symptoms like hallucinations and delusions. But the basal ganglia circuit also regulates motor behavior. It is really quite incredible. At any given time, the basal ganglia is placing tonic level of inhibitory control on the thalamocortical neurons that allow for motor behavior to happen. And when there are increases in dopamine, there's a net decrease in that control. And when there are decreases in dopamine, that control continues or increases again. And that allows for a gating of movement. And so this subtle system can become quite dysregulated when there's abnormalities in dopamine. So for example, in Parkinson's, there's too much inhibitory control. And so people are slow and rigid. In schizophrenia, there's disinhibition. So unintended movements get through. And so you can look at something like ballistic movements, jerking, withering, anything with counter contraction, like the athetosis I showed you, and understand that something is going wrong with the basal ganglia dopamine system. And so I was interested in seeing if the teenagers that met criteria for the clinical high-risk syndrome or the prodrome showed these kinds of ballistic movements. And I used a neurological side effect scale to find items that were validated for looking specifically at these kinds of dyskinetic movements. And I trained raters, and We looked for things like arm jerks, head twitches, and that kind of thing. We ruled out neurological disorders, tic disorders, and that kind of thing. And what we found was really striking. So in this particular paradigm, we recruited 40 high-risk individuals and followed them for 2 years. 10 of them developed psychotic disorders. And then we went back to baseline and we looked at the motor behavior. And what we found was the people that converted showed significantly more dyskinesia in the face and upper body, roughly the odds 3. If you had these, you were 3 times more likely to develop a psychotic disorder in a year or two. And this has been very well replicated, and our follow-up work looking at each year has suggested that the magnitude of the correlation between severity and frequency of these dyskinetic movements and positive symptoms increases significantly each year. So it's just a great at a basal ganglia dopamine abnormality without needing a scanner or PET imaging, and it's quite specific to psychosis. Cerebellar circuits are also of great interest. What ganglia circuits are involved with, selecting a particular action. The cerebellum is adding skill to that action. I like to use the example of throwing a baseball. The basal ganglia is getting you the speed and the basic thrust, and the cerebellar circuit is giving you the spin on the ball. And in addition to helping with balance, the cerebellum has gotten increased attention. It's been given the short shrift for a long time, but it's gotten a lot of good attention lately for doing a lot more than just balance and sensory integration. Work with Rick Ivory and Ito suggests that it might be an all-purpose modulator that's involved in taking incoming information and comparing it to internal models, and then updating those models based on error detection and allowing for learning and efficient behavior. And so the cerebellum is quite relevant and central to Nancy Andreassen's cognitive dysmetria model that suggests when the system goes awry, association matrices can become misaligned. There can be bottlenecks that cause slowing, and it might explain a whole range of heterogeneous psychotic symptoms. So we're interested in interrogating the cerebellum, and my lab has spent a lot of time looking at functionally relevant sub-areas. In this particular paradigm, we're interested in motor learning, and we want to interrogate these feedback and feed-forward and all-purpose modulator mechanisms I spoke of. In the old days, we would have gone to Radio Shack and bought a record player and made this pursuit rotor paradigm, but now we can use software, which is great. In this task, you trace the red cursor, it moves around in a circle. We titrate everyone until they start at the place where roughly they keep it on at about 80%. And then we repeat blocks of this over a period of an hour, and people get better at it. And we also did some cerebellar imaging, and we found that the high-risk group had smaller volumes in this relevant green area, which is more of a motor area, the anterior cerebellum, and this red Crus1 area, which is more of a cognitive area, and the superior posterior cerebellum. And so what was so interesting here was that the high-risk group didn't show the level of proficiency throughout the trial, and there was a significant difference in learning as well, and that difference was related to learning rate. And the reason why I like to highlight this work is it highlights the use of a treatment application. So, we found a specific cerebellar abnormality. We did a follow-up study where we used brain stimulation, TDCS here, with an attempt to raise the resting midbrain potential and affect motor learning. And so we replicated, the earlier experiment in an analog high-risk group, individuals showing non-clinical psychosis symptoms. And we found the same kind of thing. People with the vulnerability didn't show the same rate of learning. This is a double blind. So people got sham or stimulation and they came back, or got sham and stimulation. For purposes of illustration, I've moved all the shams to the left here. But you can see after 20 minutes of cerebellar stimulation, the groups look very similar. Currently, we're doing this work in schizophrenia with the NARSAD project, looking at it in the scanner after the TDCS session. The third circuit is one I'm very excited about, and this is the cortico motor circuit. So gesture is worthy of its own workshop. It's tied to language and development and cognition in many important ways. We're interested in schizophrenia because it might contribute to many of the social deficits that these individuals have that really contribute to disability. But we're also interested in it because many of the same areas that overlap with gesture perception and performance and interpretation are the cortical regions that we think are so interesting in schizophrenia. So when I gesture right now, for example, I'm making a round motion. That's considered a beat gesture. It's kind of like a comma. If I make like a dog while I'm talking about a dog, that's an icon, quite a literal gesture. But if I'm talking about something like love and I make a cup, that's more of an abstract metaphoric gesture. These kinds of gestures orient the listener and make them pay attention, but they also help me think and communicate. And its experimental paradigm suggests when that's disrupted on either side, communication goes way down. And what we find in psychosis is while people are psychosis risk is while people are moving more with non-purposeful movements, like fixing their collar or their hair, they're really not using these effective gestures. We also find that they're mismatching gestures more often. So they might be saying up and be pointing down, or left and be pointing right. And so there's clearly something going on there. We're also interested in the way that people perceive errors in gesture. Here's a paradigm that we're using right now in a project with Stu Shankman and Randy Auerbach, where we're asking people to look at the actor with an eye tracking study. And we're curious to see how they process mismatched gestures. The actor's going to be talking about one thing and gesturing about something else. And you'll see how disruptive this is.
This past weekend was very special because my little nephew had a birthday. Because he was turning 7, we wanted to make it an incredible event. When my mother and I had originally asked him what he wanted.
So you can see it's kind of off-putting, right? Or a little odd. You're detecting these errors. And what we're finding is that in our preliminary work, that high-risk individuals might not be detecting the errors in the same way and reacting to them in the same way. So we're developing lots of types of tasks to get at these different units of analysis and circuits. Here we see that high-risk patients are not looking at abstract gestures at the same length as controls, but they look at literal gestures, statistically roughly the same. So just briefly in the last minute or two, putting this all together, we know that people in the program might not develop psychosis. Only a third at most do. Some get better, some stay the same. And those that do develop it in different ways, quickly, not quickly, predominantly positive symptoms, predominantly negative symptoms. And so there's a lot of potential for subgrouping here. And so I've used these different motor behaviors in subgrouping studies. In this last study I'll show you, the lab looked at, we didn't look at gesture here, but we did look at basal ganglia and cerebellar markers. And using k-means, we found distinct subgroups that suggested that there were at least 3 groups of high-risk people. One of them is more defined by these basal ganglia abnormalities, one more by cerebellar, and one by neither. And these individuals have different cognitive profiles. They have different risk of conversion, and so it's really promising for getting at precision medicine or targeted treatment. And so we have a lot of hope for this kind of work. So in closing, I just want to say, one of the goals of this is to keep moving earlier and learning how to intercede earlier and earlier in the psychosis prodrome. And we are also interested in transdiagnostic perspectives. Applying this to depression is something the lab is doing in a big project right now with Stu Shankman and Sebastian Walther. We're looking at keystroke variability and motor slowing to see if that predicts remission and onset of depression. And we're trying to develop an app for clinicians to use so they can do better treatment tracking. We are building motor markers into larger studies. We're looking at the voice as a motor modulator, and in addition to that, that's influenced by the Parkinson's literature. We're seeing if we can predict the onset of psychosis by looking at dysarthria, or a type of slowing. In addition to that, we've developed scales. We have a new scale that gets at some of these RDoC units of analysis that just came out in Schizophrenia Bulletin. The lab is also looking at things like finger tapping. We try to develop skills that people can use on the computer without going into a specialty clinic, and things that really tap into mechanism. And so we have a great finger tapping task we've developed, and we're also really interested in scaling this out and looking at motor behavior much more broadly in the prodrome. One of the ways we're doing that is looking at motor behavior with instrumental approaches. Things like handwriting, for example, you can highlight everyone that you would be able to see with your eyes with a traditional observer-based method, but also highlight people that you would have missed that show subclinical movements. And so it's a really exciting development. So I want to thank my various collaborators and turn the panel over to Autumn Kajawa. Thank you so much.
Good afternoon, everyone. I am happy to be a part of this panel, and I'm going to be talking about my work on initial response to reward and the development of depressive symptoms. So if we think about the RDoC matrix, the work I'm going to focus on today is on the positive valence systems domain. And thinking about units of analysis, I tend to be towards this side of the matrix, focusing on physiology, behavior, and self-report. Positive valence systems are further subdivided into the constructs of reward responsiveness, which I'll focus on today, in particular that initial response to reward. There are also the reward learning and reward valuation constructs. So an outline for my talk today, I'm going to talk briefly about a conceptual model linking initial response to reward and depression, then thinking about how we measure reward responsiveness at the neurophysiological level and across development. And I'll show some data with low reward responsiveness as a prospective predictor of depressive symptoms across development, and then talk about what this means in terms of intervention and show some new data from my lab in terms of whether we can target reward responsiveness through interventions. So conceptually, we know that low reward responsiveness is often characteristic of depressive symptoms and of adults with depression. It's a aspect of depression that people tend to report that they experience less pleasure, less interest in seeking out previously rewarding experiences. And we also see this across units of analysis in terms of behavioral measures of low reward responsiveness and neural markers like reduced activation of the ventral striatum and other reward related brain regions. So in my work, I tend to look at depression risk from a developmental perspective and thinking about what are some of the core emotional aspects of depression that may actually precede the development of the disorder, where these things may emerge, alterations in emotional processing may actually emerge across childhood and adolescence, and they might be vulnerability markers that we can use to identify youth at high risk and potentially to target through preventive interventions. And so from this perspective, reduced reward responsiveness might actually emerge earlier. We have some evidence that we see it in offspring of depressed mothers, or at least by age 9 or so. So we might see this as an earlier emerging marker in childhood that predicts risk for depressive symptoms later on into adolescence and adulthood. But of course, we know that pathways to psychopathology are complex, and they include a range of interacting risk factors. And so I'm interested in, so we know that it's not just reward responsiveness. One construct alone is probably not going to explain the complexity of psychopathology, but considering multiple variables like reward responsiveness, how it may interact with other risk factors like parental depression and stress, both of which we know are strong risk factors for the development of depression. So in my work, I do a lot of research with EEG methods, or event-related potentials in particular, derived from the EEG signal. And so that's a way to measure reward responsiveness at the neurophysiological level that has really high temporal resolution. So it allows us to measure what's happening in the brain just 100 or 200 milliseconds after something happens. The nice thing about EEG and ERP measures also is that they're relatively economical. We can collect them from large samples. They are accessible and easily administered across development and from infants through late life, later adulthood. So the work I'm going to talk about today uses this very simple guessing reward task to measure reward responsiveness. So participants are presented with 2 doors on the screen. They guess which door has a prize behind it, and they either see an upward green arrow that indicates that they won money, or a downward red arrow indicating that they lost money. And we record the EEG signal in response to this reward and loss feedback. This task has been used extensively in childhood through adolescence and adulthood. And it's just a really simple way to measure that neural response in response following reward feedback and loss feedback. And so when we look at ERP measures, we segment that continuous EEG data in response to discrete stimuli and average across trials. And what we end up with in this reward task is we see, so here across the x-axis, time is plotted, and at 0 milliseconds is when the person is told that they receive that feedback that they won money. And on the y-axis is mean activity that in microvolts, with negative values plotted up and positive values plotted down, just for ERP conventions. So this is a neural response to gains that we see in children to feedback that they won money. And we can also look at how that averages across responses to loss. And we see overlap early on, but then around 250 to 350 milliseconds, the signals differentiate. And that is a component that we call the reward positivity. This is just showing the difference. That's a relative positivity to wins versus loss. We can also look at ERPs in terms of how they present across the scalp. And we see that this reward positivity is consistently maximal at the top of the head at frontocentral sites. And so we have a growing body of literature to support this reward positivity as a valid and reliable neurophysiological measure of reward responsiveness that can be used across development. So we've shown that it's reliably assessed across development, and it's also relatively stable across even 6 years of development, from 9-year-olds through age 15. It's also modestly correlated with observed and self-reported positive affect, which supports the validity of the measure, but it is modestly. And so that's one thing that I will mention in my talk as well, that sometimes when we look at what we think is a similar construct across units of analysis, we find that they're not actually always that strongly correlated. And it's been correlated with activation of reward-related brain regions with fMRI research, so in particular ventral striatum and medial prefrontal cortex. And one thing that I'm really interested in is that the reward positivity is elicited both in response to social and monetary reward feedback. I won't have time today to talk too much about our work in that area, but we have some really promising results coming out of my lab showing that the social reward positivity can tell us even more about the development of depression and risk and treatment. So the first study I'm going to talk about today is looking at whether low reward responsiveness prospectively predicts the development of depressive symptoms, so how reward responsiveness and depression relate to each other across time, and also thinking about what information is gained by measuring reward responsiveness across units of analysis. So this is one way the RDoC matrix has inspired me to look at these processes across different types of measures and units, and to really empirically evaluate what information is gained by integrating multiple methods. So these data are from the Stony Brook Temperament Study. This is a large longitudinal study directed by my graduate advisor, Dan Klein at Stony Brook University. So at age 3 or age 6, a large community sample of children were recruited. There's just over 600 children in the study overall. And we assessed parental depression using a semi-structured diagnostic interview with biological mothers and biological fathers when they were first enrolled in the study, and then again at age 9 when they returned for a follow-up assessment. At age 9, the children also completed that EEG reward task that I mentioned to you, and they completed a self-report measure of their depressive symptoms. And then at age 12, the children, now early adolescents, were followed up again. They completed a self-report measure of depressive symptoms again, and they also reported on their own perceptions of the individual differences in reward responsiveness using a scale adapted from the BizBest scales. And so what I wanted to look at here is whether that neural measure of reward responsiveness, the reward positivity, and that self-reported measure of reward responsiveness, the predicted depressive symptoms at age 12. And so this is controlling for symptoms at age 9 in order to look at predictors of change in that time from late childhood into early adolescence. And what we found is that both the reward positivity and self-reported reward responsiveness were associated with depressive symptoms, both such that kids who were lower in reward responsiveness showed more of an increase in depressive symptoms. And these were unique effects. So there were modest effects for each of them, but they accounted for unique variance in outcomes. One thing that was interesting and a little bit surprising here is that the reward positivity and self-reported reward responsiveness were not related to each other in the sample. So they seem to be getting at distinct aspects of reward responsiveness, but they both seem to be telling us something about vulnerability for depression. I also looked at whether parental depression moderated these effects, and we did not find effects for depression in fathers, which we've seen across a number of studies. And I'm happy to talk more about if there's questions about that. But we did find significant interactions with maternal depression, lifetime history of depression for both the neural and self-reported measure. So again, they're accounting, these interactions are accounting for unique variance in outcomes. So on this side of the slide, I've plotted the ERPs at age 9 as a function of children's depression when they were 12. And so what you can see here is if we look at the kids who were lower in depression at age 12, they show a more robust reward responsiveness, more differentiation in the response to wins compared to losses, more red across the scalp, which reflects more differentiation in the neural response to those two conditions. And then when we look at the kids who ended up having higher depression at age 12, they were more blunted. You can see less differentiation between those two conditions and less activation across the scalp. And then when we look at that moderation effect, the way that this presented is that maternal depression significantly predicted increases in depressive symptoms at age 12, as we would expect based on robust literature. But this was moderated by reward responsiveness such that maternal depression was a significant predictor for kids who were at low in the reward positivity component, and also for kids who showed an average reward positivity, average neural reward responsiveness, but it was not a significant predictor for children who were high in neural reward responsiveness. So even this established predictor of maternal depression in this age range across these 3 years was not significant for those who showed more kind of robust reward response. And the same pattern emerged for self-reported reward responsiveness, that maternal depression was a significant predictor for those at mean and low levels of self-reported reward responsiveness, but not the high levels. So this really seems to suggest that there's this underlying vulnerability reflected by low reward responsiveness that predicts increases in later depression and increasing risk across development. And we have also looked at how reward responsiveness interacts with other risk factors. We've seen that low social reward responsiveness, as measured by this reward positivity component, moderates the effects of interpersonal stress on depressive symptoms. This is in a cross-sectional sample of emerging adults. We have also, and then my colleague Brandon Goldstein with the Stony Brook temperament study data also showed that a low reward positivity interacted with stressful life events to predict depressive symptoms in that same time period from age 9 to age 12. So again, really supporting the idea that this seems to be one kind of key process involved in the development of depression. And by understanding these dimensions of emotionality and looking at them across development, we can improve our ability to predict outcomes and identify children at risk. And so for the second part of my talk, so I'm really interested in what we do with this information, how do we translate this to intervention? And in particular, is reward responsiveness a feasible intervention target with thinking about more precise approaches to treatment and prevention? Can we target reward responsiveness? And if so, how do we target it? Do our existing treatments change reward responsiveness, or do we need to tailor our treatments in some way? And so my lab has recently finished conducting an adolescent depression treatment study where we enrolled 70 adolescents between 14 and 18 years old. This pie chart shows the breakdown of their diagnoses. They were all currently depressed, and most of them had chronic depression and pretty significant, at least moderate to severe current depression. So they completed an EEG assessment before and after cognitive behavioral therapy, which was 16 sessions of group coping with depression, the adolescent coping with depression course. So a pretty intensive dose of CBT. So we had 66 complete the initial EEG, 56 were in treatment. We only had 37 that completed treatment. And then we added a second EEG a little bit later on. We lost some data due to COVID, but we had 24 who completed treatment. They completed the 16 sessions or approximately or close to it, and then completed the post-treatment EEG. This just shows the number of sessions completed, and you can see that most of them had the largest, the most common number of sessions were like 13 or 14. So they got a good dose of treatment, and these were long CBT sessions too, of about an hour and a half. And so we wanted to see, does the reward positivity increase following CBT? And we found that it does not seem to. So this is a small sample, but we're not seeing any sign of an increase. If anything, we're actually seeing a pattern where the reward positivity seems to be a little bit reduced after treatment, which we think may be due to the repeated administrations of the task in a relatively short amount of time, and maybe some lack of engagement there. And this is consistent also with my colleague, Katie Burkhouse has a paper where she looked at the reward positivity before and after CBT and antidepressant treatment in adults, and also found that there doesn't seem to be this overall increase. So CBT, traditional CBT, is not really enhancing reward responsiveness. But what we did find here is that relative increases in the reward positivity are associated with reductions in symptoms of depression, and more specifically, symptoms of anhedonia. So this is looking at change from pre, minus, post-treatment reward positivity and anhedonia. So the teens who are up here on anhedonia showed more of a reduction. The ones who are down here in reward positivity showed more of a relative increase in the reward positivity at time 2. And you can see that those who showed a relative increase showed also that related to more of a reduction in symptoms. So it's a small sample, and so there's some limitations here, of course, but this tells us that something about moving around reward responsiveness does seem to have clinical utility and to be clinically meaningful. And so lastly, my lab, we've been really interested in how we can refine interventions to more directly target the reward positivity and reward responsiveness. And so we conducted a proof of concept or analog study to see if we are able to kind of move this around in a lab session through a brief motivational manipulation. And we found that we are actually, with instructions, manipulations more directly targeting reward responsiveness in the session, we're able to kind of modulate reward positivity. So it does seem to be something that could be an intervention target. So what we did here is we had 98 emerging adults complete an ERP monetary incentive delay task, and they completed it 2 times. The first time, everyone got standard instructions on how the task works. The second time, they were randomized to the neutral standard instruction again, but a little bit longer, or a brief motivational manipulation. And so for that manipulation, we prompted participants to think about what it is that they want to do with their earnings, and kind of think about a personally salient reward and why they want to earn that, and to think about how much they want to win those rewards. And so we found that there were no differences between the groups in reward responsiveness in round 1, but there were significant effects of this motivational manipulation in round 2 across units of analysis when looking at reward responsiveness. So in round 2, participants in the motivational manipulation reported increased motivation to win. They also showed faster reaction time on targets in order to obtain a reward, and they showed an enhanced reward positivity ERP component. And so you can see that here, the motivation group. So this is looking at win versus loss with more positive values reflecting greater differentiation, or with the red values here reflecting more differentiation. So we see more of a reward response in that motivational group compared to the neutral group with the second administration. And looking at the ERPs here with this solid green line, you can see more of that's the motivation group when they win money in that second round. And you can see more of a positivity in the motivation group compared to the neutral group. So pulling this all together, these data suggest that low reward responsiveness seems to be an underlying vulnerability for depressive symptoms. And so studying it across time can help to inform how symptoms emerge across development. And it seems to prospectively predict increases in depressive symptoms and moderate the effects of other risk factors on outcomes for children and adolescents. these data also suggest that we get unique information across units of analysis with the RDoC matrix. And so there's some challenges with looking at multiple levels of analysis because sometimes they don't always relate to each other in the ways that we think they should, or they're only modestly associated with each other. But these data support the utility of integrating information from multiple measures, even if they're not telling us, they're not getting at the exact same process, they're actually improving our ability to kind of predict outcomes and to account for more variance in outcomes. And so the reward positivity does not seem to increase following our standard cognitive behavioral therapy for depression, though. So that suggests to me, but relative increases are correlated with reductions in anhedonia. So it suggests that there is some clinical utility to moving reward responsiveness, but we have to figure out a better way to directly target reward responsiveness. And I think that's a really important next step in terms of precision or personalized approaches to intervention. And so one way, it does seem that it's modifiable through manipulations that more directly target reward salience and motivation. And my lab is working now to figure out what does this mean for prevention and whether we can then use some of these approaches to increase reward responsiveness earlier in children at risk for depression. So I would like to thank my lab, the Mood, Emotion, and Development Lab at Vanderbilt, and also my graduate mentors, Dan Klein and Greg Hychuk in the Stony Brook Temperament Study. And our next speaker is Dr. Anne-Marie McNamara at Texas A&M.
Hi everyone, I'm Anne-Marie McNamara. I'm at Texas A&M, and I'm very happy to be part of this, and I want to thank APS and the RDoC team for inviting me to participate. So I'll be talking today about our work looking at the effective neuroscience of anxiety. If I can get my slides to advance, just a second. There we go, okay. All right. So the measures that I'll be talking about in terms of the RDoC matrix fit under the negative valence system domain. And first I'll start by talking to you about some evidence that we have showing that anxiety disorders are associated with increased attention to negative stimuli. And then some work we found which shows something of the opposite, which is that some patients might be characterized actually by blunted responding to negative stimuli, that is reduced attention to negative stimuli. Then I'll move on to some of our more recent work, which was inspired by the RDoC initiative. And I'll present you some evidence of an anxiety spectrum, as well as a candidate brain profile of comorbidity load that might signal increased symptoms and worse prognosis in the anxiety disorders. So anxiety is costly on both a personal and societal level. It is prevalent, and it is principally diagnosed by clinical interview. We have no neurobiological or objective tests to diagnose anxiety. Current treatments are only moderately effective, and treatment decisions are made based solely on provider and patient preference. We have more than one treatment. Some work for some individuals, some work for different individuals, but we have no way of sorting out which treatment is best suited to which individual. If we had a better understanding of the pathophysiology of anxiety, we might be able to develop new treatments that work better and target these aberrant pathophysiologies, but we also might be able to better match patients to the treatments that are best suited to them. One candidate mechanism underlying anxiety is excessive negative emotion generation. And I'm going to define emotion here just so we're all on the same page. So when I say emotion, I'm referring to a multi-layered, coordinated response to a stimulus that an individual appraises as having relevance to its goals. Or in another way of saying this, it would be an action tendency. And so what do I mean by that? Well, if we're walking down a dark alley at about 2 or 3 a.m., we might have certain emotional responses. We might feel afraid, we might feel our heart pounding, we might have these action tendencies which prepare us to fight or to flee. So we are prepared to engage in the actions that would benefit us in this situation, and our attention is drawn to the potential threat of the situation. So evolutionarily, emotions have done us well. They have saved us. But when our attention to negative stimuli or our emotional response is excessive, or when there is no real threat, but we have those same kinds of emotional responses, that's when it is no longer protective, and it can actually become quite debilitating. And that's what's thought to be happening in anxiety, that basically these systems, which have generally served us well, are sort of hijacked and are over-functioning and impairing us rather than aiding us. In the lab, we can study emotion using event-related potentials, or ERPs. And Autumn has already introduced you to these, so I'm not going to spend a lot of time on this. But just briefly, in the context of studying emotion, we are typically showing participants emotional and neutral stimuli and comparing their brain responses to these different types of stimuli. So this event of interest at 0 milliseconds would be often the presentation of an emotional or a neutral picture. And following that event, we see a cascade of brain responses, which we can record in the ERP. ERPs have high temporal resolution, and for this reason, they are complementary to fMRI. And here's a schematic of the typical type of experimental setup that we might have. So we have a participant in the top left-hand corner there, and that person is viewing a negative picture. And then we have other pictures that we show them, some of which are negative and some of which are neutral. And we're recording their brain activity using EEG throughout this time. We're sending triggers to that EEG data so that later we can pull out the brain activity associated with one kind of stimulus versus another. We can average across trials, and we can produce ERPs. The ERP that I'll be focusing on today is called the late positive potential, or the LPP. You can see it depicted here. And as in Autumn's graph, positive is down just by ERP convention. So unpleasant pictures, which are denoted by the black line, are eliciting larger amplitudes when compared to neutral pictures, which are denoted by the dashed line. That positivity is maximal at centroparietal sites, as you can see on the head. So the LPP is larger for emotional compared to neutral stimuli. It's thought to measure the elaborated processing of stimulus salience. And it receives contributions from a wide variety of neural generators, including both limbic regions and prefrontal regions. When we have used the LPP to test this idea that anxiety should be associated with increased attention to negative stimuli, we have in fact confirmed that hypothesis. So the data in A there on the left is an unselected sample, and on the bottom on the x-axis is state anxiety. And we see it as state anxiety increases. We have increased LPPs for negative stimuli. We also see this in GAD, generalized anxiety disorder. That's depicted on the right. So these are clinical individuals, and they also show larger LPPs to negative stimuli. Yet in contrast to this work, there's also the idea that some forms of affective psychopathology might actually be characterized by something of the opposite, and that is emotional blunting. So this would be smaller responses to negative and positive stimuli compared to neutral stimuli. And the notion here is that maybe this is actually an evolutionarily conserved response to adversity. So when things get bad enough for long enough, it might be adaptive at some point to simply turn inwards and to stop attending to external stimuli, to stop trying, essentially, stop seeking out or attending to these stimuli. And this would serve to conserve valuable energy resources when efforts and attempts to change situation have been futile in the recent past. And there is evidence of this emotional blunting, primarily so far in depression. This comes from the emotional context insensitivity work. And there is evidence that depression is associated with reduced attention to a variety of negative stimuli and positive stimuli using a number of methods. So we set out to determine whether that enhanced LPP that we saw in GAD might be different if these individuals had a comorbid diagnosis of depression. And as pointed out by Sarah Morris in the beginning, this is a very relevant question because a lot of individuals are comorbid when it comes to all kinds of psychopathology, but particularly GAD is especially known to be comorbid. So a lot of these individuals in the natural world, when not selected to be just with GAD, would have a diagnosis of comorbid depression. And so in this study, we had individuals with pure GAD who just had GAD, and they did show the heightened LPPs to negative stimuli. But then we also had individuals who had a comorbid diagnosis of depression, as well as a pure MDD group. And once we add depression in there, we no longer see that heightened response to negative stimuli. In fact, we see blunted response when compared to controls or those with GAD. So this suggests that the presence of a comorbidity like depression might change that pathophysiology. We might see something different going on in these individuals with more than one diagnosis. We see similar results in youth. So this work was led by Autumn Kujawa. And what we see here is a youth sample with anxiety disorders and then youth healthy controls. And we see that when we look at the LPP in these individuals, it is larger in the anxiety disordered group compared to controls. So this is in line with the first results I showed you. It's the more basic idea that anxiety should be associated with increased attention to negative stimuli. So we do see that at the group level. But when we look across individuals in terms of their differences in depressive symptoms, we actually see something of the opposite. So these are the same individuals, same stimuli, and now we see that those who have more depressive symptoms show smaller LPPs to negative stimuli. So this suggests again that there might be more than one process here and that there might be sort of group differences in terms of heightened LPPs, but that levels of comorbid depression, maybe increased severity, greater distress symptoms, might be characterized by blunted responding to negative stimuli. So far I've shown you some evidence that anxiety is associated with increased attention towards negative stimuli and that we can measure this using the LPP. And I've also shown you that there might be some individuals in there who actually show evidence of blunting. This might be linked to comorbidity. In this next study that I'm going to present, I'm going to show some of our work that has started to look across anxiety disorders with the ultimate aim of refining nosology. And in line with the RDoC initiative, we have been trying to integrate multiple methodologies to achieve a more systemic understanding of negative emotion generation across anxiety. So comorbidity in the anxiety disorders is the norm. At least 50% of individuals at any given point in time will have another disorder. And comorbidity load is clinically meaningful. It's associated with greater symptom severity, increased disability, higher risk of suicide, less chance that these folks will do well, that they will actually recover from their illness. And if they do, it's going to take them longer and more resources to get there. So as a clinician, if a patient walks into my office and I know they have more than one diagnosis, this should signal something to me. This is prognostic. This should tell me that this person has more going on and, importantly, that they're not as likely to do as well in the future. And yet, I don't know what to do with that information. I am likely to give them piecemeal treatment to treat one or maybe two of their diagnoses. I don't have a treatment for comorbidity. I don't have a special extra treatment that I give them. And that's largely because we don't understand the pathophysiology of comorbidity load. We know it's clinically meaningful, but we don't have any ways of tailoring treatment to these individuals. So perhaps if we had a better understanding of the pathophysiology of comorbidity, we would be able to provide better clinical care to these individuals. So the work that I'm about to show you uses ERPs, but it also uses fMRI BOLD, which measures changes in blood flow that are associated with neuronal activity. In complement to ERPs, it can achieve submillimeter resolution, so it has a very high spatial resolution. And in terms of BOLD, what would we expect when it comes to anxiety and depression? What would we expect to see? Well, in brief, when we're looking at negative pictures, we would probably expect to see heightened activity in the salience network. The salience network is comprised of the amygdala, the insula, and the anterior cingulate cortex. And it is involved in detecting salient stimuli in the environment and sort of alerting the organism to those stimuli and sorting out priorities in terms of attention and action. And so when viewing negative stimuli, we would expect increased activation in this network. So this data that I'm about to show you was recruited in a way different to our prior studies. And what we did here is we focused on comorbidity load. So we wanted to achieve a range of severity across a sample of anxiety disordered patients. We required that all patients had a focal fear diagnosis, by which I mean specific phobia or performance-only social anxiety. And we required that because we wanted to avoid the confound that might otherwise occur, whereby those who were more comorbid or more distressed might be absent of that focal fear symptom. So we didn't want a situation where the low comorbid individuals had lots of focal fear and then the high comorbid individuals were simply distress-oriented and just didn't have that focal fear core. So we wanted to hold that constant across the sample and then allow comorbidity load to vary on top of that. So we had two groups. We had a low comorbid group who had a focal fear diagnosis and either 0 or 1 other diagnosis, and then a high comorbid group who had, again, that focal fear core. and then 2 or more other diagnoses. All participants performed a passive picture viewing task. They viewed negative and neutral pictures, and they had an fMRI recording and an EEG recording in separate sessions. And these are our results. So we found that individuals who were more comorbid had greater amygdala activation to negative versus neutral stimuli when compared to those that were less comorbid. We also saw smaller LPPs. So in these same individuals to the same stimuli, we saw that those who were more comorbid had smaller LPPs compared to those who were less comorbid. So together, this suggests a neurobiological pattern associated with comorbidity load that might be comprised of heightened alarm as signaled by greater salience network activation, along with reduced motivated elaborative processing as indicated by the blunted or reduced LPPs. We know that elaborative processing is probably necessary to bring about changes in the fear structure. So this is the basis of exposure therapy, right? We take individuals and we get them to sit with those anxiety-inducing stimuli until they can receive information that contrasts that fear structure and can bring about changes in the level of threat or salience they ascribe to those stimuli. So by sitting with the stimuli and allowing that elaborative processing to occur, the change happens. Without that elaborative processing, as indicated by the LPP, we might have a cyclical phenomena here where individuals who are more comorbid have that heightened alarm response, but they are unable to bring about changes in the salience of that stimuli because they never fully engage in that more elaborative prefrontally mediated processing. Therefore, this profile, which we're referring to as harm-A, might be a candidate unifying mechanism by which anxiety is maintained and worsens over time with individuals picking up additional diagnoses as they proceed. I'm going to show you some evidence that has supported this idea so far. So this is in the same sample. I'm just going to approach the question from a few different ways. And so what I'm going to show you here will be laid out along an amygdala-insula connectivity dimension and a LPP dimension. So what we found was an interaction between amygdala-insula connectivity, so that's the salience network, and the LPP. This was found in a continuous fashion across these patients, but I'm going to show you some data broken up into groups here just because it's easier to visualize those brain responses when we split people up into quadrants. So the first quadrant I'm going to show you are individuals who had high levels of amygdala-insula connectivity as well as small LPPs. So these are the harm-A folks. Those are these individuals with this heightened alarm and reduced motivated attention. And you can see that heightened alarm in the insula connectivity in that brain, and you can see that smaller LPP at the bottom. These individuals had high scores on the latent depression measure. So they had high levels of latent depression. So these are our group that we would expect to have high levels of symptoms, and they do. Every other quadrant, so the other 3 quadrants, did not show this high level of depressive symptomatology. And so I'm making them big here just so you can see what's going on, and then I'll send them to their respective quadrants. So these individuals had low levels of amygdala-insula connectivity and low or small LPPs, and you can see that in the brain and in the LPP waveforms. And you can also see their depression scores. They did not have elevated depression scores. This next quadrant, these are individuals with low levels of amygdala-insula connectivity, but high or large LPPs, and they also did not show high levels of latent depression. And finally, individuals in that final quadrant who have both high levels of amygdala-insula connectivity as well as large LPPs, they also do not show those high levels of latent depression. So it's really these folks in the top right-hand quadrant, those with the harm-A profile, that show the high levels of latent depression. We were also interested in knowing whether harm-A might be related to historical comorbidity load. So we have this working hypothesis that maybe harm-A sort of cycles upward with pathology. So there might be this sort of scar phenomenon by which bouts of psychopathology, prior historical diagnoses, might increase harm-A, which might in turn increase further lability for psychopathology, so that these individuals sort of spiral upwards in terms of accruing additional diagnoses, becoming more comorbid, more severe, more debilitated over time. And so we used harm-A to predict, using a logistic regression, whether individuals were more likely to fall into a low or a high historical comorbidity load group, so based on the number of prior diagnoses. And what we found was in line with the notion that harm A is increased by historical comorbidity load. So individuals with high levels of amygdala activity, as indicated by rightward side of that amygdala graph on the x-axis, and low LPPs, that's the red line, or small LPPs, these are the harm A individuals. And you can see that they have a higher probability of having had a high historical comorbidity load. So these are the individuals with more historical diagnoses. And I should say that this is controlling for the number of current diagnoses. Finally, we did have the opportunity, thanks to COVID actually, to recently connect again with these individuals 12 to 18 months after their initial brain scans and to see how they were doing. And so we have time 2 data on these individuals, and we were able to see whether this harm A brain profile predicted worse symptomatology at time 2, controlling for time 1. And this is specific to dysphoria here, what I'm showing you. And what we see again is that higher harm A at baseline at time 1 is predictive of greater dysphoric levels at time 2. So it's the same pattern here. We have that low or small LPP with the red line, and then we have the high levels of amygdala activity on the x-axis at the right of each of those graphs. So it's present for both left and right amygdala. So again, these individuals with high harm A are the ones who have higher time 2 dysphoria, controlling for time 1 dysphoria. So all in all, we have this brain profile, which we think is predicting future symptomatology, which relates to current symptomatology, and which appears to be also related to historical symptomatology, suggesting that it might track comorbidity and the worst prognosis associated with comorbidity load across the anxiety spectrum. So in sum, I've shown you that anxiety is characterized by increased attention to negative stimuli. I've shown you at the same time that there is some evidence of blunting. And in trying to explore how those 2 mechanisms might fit together, I've shown you evidence of an anxiety spectrum and an EEG fMRI brain profile that might mark comorbidity load and indicate worse prognosis across the anxiety disorders. In terms of future directions, I think that it will be important to sort out whether blunting is related specifically to depression, or whether it might be something a bit broader than that, related perhaps to distress in general, or diffuse anxiety. I also think we need to revisit the RDoC negative valence systems and take a look at whether we might want to sort of try to split that up a little bit more, or think about the different multiple processes or stages that might be involved in negative valence systems. We have here in the work I've shown you 2 measures of negative valence system activity, but they're at the same level. So they're both brain measures, the LPP and the fMRI BOLD, and yet they're telling us different things. And so I think it's going to be important at some point to sort out whether even at the same level of analysis, we might have different processes that can be measured by different measures. I want to thank my lab, the Multi-Method Affect and Cognition Lab, my grad students, mentors, and collaborators, and NIH for funding this work. Oh, sorry. I am forgetting to introduce Dr. Uma Vaidyanathan, who is going to sum some of this up for us. Uma's from NIH.
All right. Thank you, Anne-Marie. Thank you for the wonderful talk as well. Hello, everyone. I am going to go through my slides quickly since I know we are running short in time, and I would like to be mindful of your time. So we may not have time for a full-fledged Q&A session today, but what we'll do is have all our various presenters try to respond to your questions offline via email or some such. We'll check with Andy about that. But anyway, it's lovely to be here with you today, and I'll try to sum this all up in a nice way. What I'm going to do in the next few minutes is go very briefly over our talks, highlight common RDoC relevant themes amongst them, and then focus on sort of questions or challenges that our various panelists highlighted. So here we go. Again, great set of talks. Vijay started off today by discussing the role of motor abnormalities in predicting conversion of clinical high-risk individuals to psychosis. Autumn then picked up on that developmental theme, but she moved the spotlight to using reward responsivity in predicting depression. And then Anne-Marie focused on using MRI and EEG to parse depression and anxiety and see what they had in common versus unique aspects. There were numerous themes amongst these studies that exemplified the principles we at RDoC advocate. And I've listed a few on these slides here. And to be clear, these principles, a lot of these were run before RDoC was formally launched, and they've been advocated by others in the field as well. So the goal of RDoC is not to claim that we were the first to do it, but rather, as Sarah said, to put it all together in a package and as an initiative, as a set of research strategies that researchers can use in a convenient way when designing their studies, especially because researchers had to use some of these things in really convoluted ways pre-RDoC. So the first and most obvious theme you saw among studies was the focus on constructs rather than just categorical diagnoses in all of them. So Vijay's study looked at clinical high-risk individuals, automated reward processing, and Anne-Marie on depression, anxiety, and their intersection. So while our researchers used some sort of diagnosis starting points, they did not focus solely on these labels. The second theme was, again, the dimensional aspect. So our researchers used a lot of dimensional measures to study these, which is advantageous both from a clinical perspective, understanding how these phenomena manifest in the real world. I mean, people aren't just anxious or not anxious, they have a spectrum, right? And the other aspect is statistically, it gives you more power for your study when you do that. So that was the second major theme. The third major theme that you saw was the use of multiple methods, such as EEG, MRI, and self-report. Now, this is a particularly important theme for us because, as Sarah mentioned, to date, most mental health diagnoses and phenomena are studied using self-report or other report of feelings and behaviors. And while this is a useful start, going beyond this to fully understand the connections between brain, body, environment, and development is important. And that's why we at RDoC focus particularly on mechanisms. And finally, one of the critiques we heard every now and then at RDoC is that it ignores the developmental component to mental disorders. And as you can see, Vijay alluded to this, but then we have tried to expand upon this, and our panelists have clearly focused on studies that have a strong developmental angle to them. So we would like to say this is really not the case, and that RDoC is in fact very amenable to developmental studies. At this point, I think, given our time limits, what I will do is skip over the rest of my slides and hand it off to Andy to wrap up. Andy, did you want to take over here since we're at 5 o'clock already?
Absolutely, that sounds good, and we do want to be respectful of everyone's time. What we'll do is we'll brainstorm a way to address everyone's questions and have them responded to by our participants today in a great way. Look forward to a follow-up email if you registered for this webinar to learn more about how we can arrange for that. But bottom line, we're so thankful to our presenters from the university community and from National Institute of Mental Health for participating today. I'd also like to thank colleagues from NIMH, Jenny Pacheco, Syed Rizvi, and RDoC unit head, Bruce Cuthbert, who's on the line today. And from the APS office, my colleague Kekoa Erber, Michelle Johnfin, and Nick Antuch for their help today. And thank you very much to our audience. We really appreciate your participation. Again, we'll be making portions of this recording available online following this webinar, and we'll be in touch with you about ways that we can continue a discussion based off of the wonderful content of the scientific talks we heard today. Thank you very much. I appreciate it so much, and we will see you again soon. Have a wonderful day.